All right, welcome everybody. Thanks for being here. It's nice to see you all. Welcome to USA Blue Schools webinars. We've had a number of them this year, and it's been great to allow some of our students, especially, and our educators and our partners to meet some scientists and some other marine professionals. So we're really glad you're here. If you're not on our mailing list, I'm going to put the link to that in the chat in a couple minutes. Please do get on our Blue Schools mailing list so that you'll hear about all of our events and all of the information that you're missing out on. So we are so glad to have our speaker today, and I'm going to let Tammy introduce her.
Hi, everybody. Tammy Lunsford, and I am thrilled to have with us today Dr. Aditee Mitra. Dr. Mitra is from Cardiff University in the United Kingdom, and she is going to share with us a little bit about mixoplankton. I was able to see her talk while I was at the Ocean Sciences meeting in Scotland and was truly blown away by both the science and the exciting way that she presents a really revolutionary type of science compared to what most of us teach. She's been she has so many publications that I actually lost track. I have no idea if you even have a number of how many publications that you have related to her work in the ecology and evolution of mixoplankton. So we are thrilled to have you, Dr. Mitra. Welcome, welcome. It's all yours.
Thank you so much. That's very kind. And I shall start with sharing my screen, and hopefully it all goes to plan. And you can see it.
Looks perfect.
Excellent. Okay, so thank you very much. Thank you, Tammy. Thank you, Megan, for inviting me to this webinar. It's really exciting. And thank you, Tammy, for your very kind words. I'm excited about mixoplankton, so I hope I can share the story of mixoplankton and get you all excited about it as well. So to start off with, I'll go through a few terminologies, and it's basically because I'll refer to them quite often, so it's probably easier for me to remember what I'm going to say, for you to also be on the same page as me. So we start with a microbe, which is a very small living organism that can only be seen with a microscope, hence the microbe, and usually it refers to single-celled organisms. It is also a generic word which is used for a diverse range of organisms, from bacteria, fungi to protists. Then we have plankton, so that's from the mixoplankton as well. So these are organisms that drift in the water. They cannot swim against water currents or tides. So they can swim up and down in the water column, but they won't be able to swim against the tide. And this is why when you go to the sea beaches, you'll find the sea jellies all stuck in the beach, because they have come with the tide and they haven't been able to swim out. And an important component of this plankton, the marine plankton, are protists. So that's our next word. So the protists, this is a generic term. It's really a bucket for lots of different organisms. These are single-celled eukaryotes. So that's one definition. But then, so it means they're not bacteria, they're not cyanobacteria. So that's a blue-green algae. So some of you may see them as the scum on ponds in the summer months. We do here. Fungi, plants, or animals. So these are not protists. And then mixoplankton, what are these? So mixoplankton are microbial protists. So they're eukaryote, single-celled organisms. And they are so they're plankton, and they're mixing different contrasting trophic strategies, which are not seen in any other plankton, and hence the term mixoplankton. So the term was coined in 2019, and hopefully by the end of this talk, you'll see why we felt we needed a new term. So these mixoplankton they photosynthesize, and they also eat prey. So with those trophic strategies from the mixoplankton, as I mentioned, so the different protist types have different trophic strategies. So we've got osmotrophy. So one of the things I'm going to use are these hexagons, different colored hexagons. So they will be for the 3 different trophies. So just keep an eye on it, and I'll have it up and down. So that's what it's doing here. So osmotrophy is the use of dissolved organics. So it potentially, if there's enough dissolved organics, it can provide all the resources required by the protists to support growth, the organism growth. Then we have phototrophy. So this is photosynthesis. They use light and inorganic nutrients, and it provides all the resources, but only after complete synthesis of all the intermediaries. So they're from the inorganic. So there's a cost associated with this. And then there's phagotrophy. So this is the consumption of food. So this is eating prey. And this is, again, this can provide all the resources. Well, look at us, we don't photosynthesize. So, but the problem is it has also got a high loss rate. So this is with voiding and with SDA, which is excretion respiration. And these are the 3 main strategies which we will look at today. And with the phagotrophy, there is the interesting bit with the mixoplankton is when it couples phototrophy and phagotrophy, then it can actually address the loss, and it could make it through internal recycling. It can use the end product of phototrophy to drive phagotrophy and vice versa. So that's that. So food webs. So we've got the trophic strategies. What does it mean in our food webs? So we've got over the centuries, the marine food web has been very much mirrored again, based on the terrestrial food web. So you have got at the base the primary producers, then you've got the primary consumers, secondary consumers, and the tertiary consumers. You have got all the waste product going into decomposers and the nutrients then driving the whole cycle. So similarly, when we look at the marine food web, we find at the bottom the primary phytoplankton, which are the primary producers, the protozooplankton, the primary consumers. Then we have got the metazoans. So these are mesozooplankton animals. So you've got the jellies, the crabs and copepods, shrimp, etc. And then you've got the tertiary ones, so the higher trophic levels. We've got the decomposers and the nutrients and the whole cycle happening. And in this food web, at the base, we have the microbial plankton. So these are the cyanobacteria, which are not eukaryote. So they're the prokaryotes and the protists, which we have all the mixoplankton, phytoplankton, etc. in. So the traditional concept of the protist plankton types has been that we have got phytoplankton, which are the primary producers. So here the phytoplankton we have got, it takes up dissolved organics through osmotrophy, and it uses uses light and nutrients to do carbon fixation. And then at the other end of the spectrum, we have got the primary consumers, which are the zooplankton, protozooplankton, which eat the primary producers, the phytoplankton. And here again, we have got the osmotrophy. It does also the osmotrophy, so it takes up dissolved organics, and it eats. So osmotrophy is one of the ancient traits. So this plant-animal dichotomy has been at the base of marine science forever, and it's in textbooks, in climate change and fisheries models, and everywhere you see. So how did this happen? We were interested to find out how did it evolve from, which one came where and first. So we have the phytoplankton, which are doing the osmotrophy and phototrophy, and the zooplankton, which are doing osmotrophy and phagotrophy. So which one came first? We assumed, like most people, usually everybody will assume that the plant will come first and then the animals. But this is not how it happened. It was actually the protozooplankton is the ancestral form in the protist evolution, plankton evolution. So this then, at some point over millennia, the protozooplankton would have evolved to gain phototrophy. So it gains a phototrophy. And then again over evolution, millennia, it loses the ability to eat and it becomes a phytoplankton. So instead of going from plant to animal, we actually in the evolutionary process, we find it's gone from animal to animal-like zooplankton to plant-like phytoplankton. So once we found this out, we wanted to find out how did it happen and what happened to the mixoplankton, because they are not there in the books. We didn't do mixoplankton or anything. So why have they gone missing and where are they in the evolutionary line? So what the pathway is, what we believe is that we've got the ancestral zooplankton, which would be taking up dissolved organics and eating prey. So they would have had cyanobacteria, originally would have been eating. And at some point during the millennia, it would have evolved to take the cyanobacteria, their prey, and keep the plastids, the chloroplast from this prey. They would acquire the phototrophic capability. So they would have stolen the chloroplast from the prey and kept it inside them. And then they became the mixoplankton, and where the photosynthesis has to be acquired. So in these types of mixoplankton, we have got phototrophy, which is not inherent, so they always have to eat. And because it's not inherent, because they have to constantly take in prey to acquire capability to photosynthesize, we call these the non-constitutive mixoplankton. And then we wanted to know how many of these types are there. So we looked at how were they taking in their prey, how were they stealing, which part of the body parts they were stealing, what were they keeping, how were they doing, and we found there are actually 3 types of way they acquired their phototrophic capability. So first up, we have got the generalist non-constitutive mixoplankton. So the generalists are, as the name suggests, they take food from generic prey. They take it from a diverse range of prey types. And because they take from such a diverse range of prey, they haven't evolved the capability to look after their stolen chloroplasts. So they'll take these chloroplast from the prey, they'll keep it and use it, and they need to acquire these regularly, daily, nearly daily. So we've got an example, well-known example of Strombedium, it's a genus which has lots of species. So you can see the green things are their acquired chloroplasts. And then we have got Loboya, which it looks like an ice cream cone, but with all the different, again, the green bits are the chloroplasts. So what would have happened is, because they are also eating and they're stealing the chloroplast, they could have potentially in the past been confused with very well-fed protozooplankton, because the zooplankton also put the food into a digestive vacuole and takes time to digest it. And at the time you would see that they have got the color there as well. And these things would be stealing and keeping them for photosynthesizing. So that is why perhaps they got missed out. And the good thing about this, and they are very good fish food. The fish absolutely love these GNCM. And we got a video here from George McManus, works in Yukon, and you can see, so this is a seaweed, ulva, and you can see the Strombedium. So what they're doing, they're stealing chloroplast from the swarmers of the seaweed. So that's the diversity in where they can get their chloroplast from. So it's not just something smaller than them. They can take it from something this big, with a seaweed swarmer. So so you can see all these little things are Strombedium going and stealing their chloroplasts. So we have the first set, then we have next. So the 3 modes. So now we have got the plastidic specialist. So as soon as you see the word specialist, there's something special about them. The specialty is they need specific prey to be able to acquire the phototrophic capability. And because they get it from specific prey, they have also evolved the capability to look after these stolen parts for a few weeks. So here we have an example. This is a Dinophysis, which is, we've got here again, osmotrophy and phagotrophy. So it doesn't have its chloroplasts. These are important organisms in the environment. They need the chloroplast from these cryptophytes, which are called teleolax, which are as you can see, they're very small, and they cannot steal the chloroplast. They cannot eat teleolax because of the size difference. So we enter thief number 1. So this is another specialist NCM, which is now a ciliate. So you've got 3 different types and taxonomically very different groups. So this mesodynium needs to steal the chloroplast as well from the teleolac. So the mesodynium steals the chloroplast, eats the teleolac, steals the chloroplast, some nuclear material, keeps it in their body uses it to do phototrophy. And then we have Dinophysis, which goes and steals it from the mesodynium. So these complex, they're very much dependent on each other. And here we have a video of a Dinophysis. So you can see this is a Dinophysis. This is a mesodynium, and there in the background, the circular thing. So you can see the size difference between a Dinophysis and a mesodynium. So teleorex is smaller again. So what the Dinophysis does, it uses a harpoon-like structure to capture the PSNCM, the mesodynium, and then it puts in a peduncle, which is a drinking straw-like structure, into the mesodynium, the prey, and it uses is suck the innards out of the prey. And then what it does, it keeps a chloroplast and digests the rest of the mesodynium. So let's start. Let's start the video. Yeah, there we go. So you can see it's made, you can see the peduncle. Keep going. There, you can see there's the straw-like peduncle, and you can see that the mesodynium body parts are gradually being sucked through this peduncle into the Dinophysis. So basically both of these organisms need the specific teleolacs, and because it's only keeping the plastids or the nuclear material, so we named it the plastid. Well, we use the term plastidic specialist non-constitutive mixoplankton. Oh, okay. Just doing it again. Better. Right, so then we have number 3. So we got the ones which were generalist, we got the specialists, which were only stealing plastids and nuclear material. Now we've got specialists, which acquire phototrophy by taking in the prey and maintaining them as symbionts inside their cell. So we're still talking about a single-celled organism. So we still have the single cell. It's got the ability to take up dissolved inorganics. It also takes up prey to digest. But some of the prey, and we're not sure why and how it chooses which one, but some prey types it will take in and maintain as symbionts and use those to do the phototrophy. And examples of these include some very important biogeochemical groups, which you may have heard of. You've seen the forams, which are everywhere in the oceans, and the radiolarians. So we have another video here. Okay, and let's see. Okay, so here we have got a foram in the background. This is an Artemia or a sea monkey. We call them sea monkeys here. I don't know if you call the same thing, but it's very important food in aquaculture. So these tendril-like things are the foram with its not tentacles, but it's the foram spicules which are extending out. So it's captured the Artemia and it's gradually dragging it inside. You can see it's gradually going in, taking it in. It's going to take it into this is where the body is. It's going to take it in and it's going to digest it there. And when Professor Spiro changes his focus, you can see if you see these little dots here, these are the symbionts. So what it does, it has a symbionts on these, like thread-like. You can see them now and then. Let's see. So there, because that's better. there. So you can see the symbionts here. So what it does, it has the symbionts. The symbionts, they sort of send them out on these thread-like structures out during the daytime. So it does a photosynthesis, and again at night, then it will retract them back in. So this is the same sort of thing with the radiolarians. So these are the rhizarians as a bigger group which do it. So it's eating and photosynthesizing. But it's clearly, these are much smaller, the symbionts are much smaller than the prey it's eating to digest. So we go back to our evolutionary pathway, and we have got our 3 types of non-constitutive mixoplankton, which are acquiring phototrophic capability by stealing prey or keeping them as symbionts. Then at some point in the millennia, the evolution went on happening, and the mixoplankton evolved the capability to maintain the chloroplasts for photosynthesis. So it then put it in its genetic code, in essence, to be able to maintain, and keep going and go on through the generation. So it's now it's not requiring to top up, it's got it in itself. So these, because it's got an inherent capability to do the photosynthesis, that's why we call it constitutive mixoplankton. And there are a lot of examples in this. So here we have got example of 2. We have got a tripos, which is this organism here, and then a smaller one, which is a fragilidium. And again, I think we expected the big one to be the small one, right? So let's see. So we've got them here. There you go. So individually, they both are constitutive mixoplankton. So they both have the ability to eat and to photosynthesize. But here you can see the fragilidium are actually eating, 3 of them are eating the tripos, which is another mixoplankton. So they keep going, keep fighting over the bits as much as they can get. And if I watched this video so many times that I can still watch it. So this has been done by Dr. Urban Tillman from RV, and Urban makes absolutely amazing videos. So there you go. So it's eaten and gone. And this sort of thing then changes our idea of how the food pyramid works, because usually it's assumed that small things are eaten by big things and bigger, etc. So now we've got small things eating bigger things. So it's not just the fact they are doing both the things, they're also changing our view of how the pyramid is working. So we're back to this evolutionary pathway. So obviously I have not finished with this. So what happened then? We have in evolutionary terms, then at some point, they lost the ability to eat. They lost the ability to hunt and consume prey. And this is where we have our protist phytoplankton. So instead of the phytoplankton, which has been always assumed to be at the base, we have the phytoplankton, which came right at the end of the line, so to say. And also this evolution has not been in one way. It's not just a linear direction. Over millennia, there's been lots of evidence of the GNCM, the non-constitutive mixoplankton, going back to protozooplankton and going back here towards another non-constitutive, and CMs going. What we have not seen yet are any protist phytoplankton going back and then this whole thing happening. But between these 3 forms, there's been a lot of going forwards and backwards. And we shall see the implications later on in the tree of life. So when we go to, so what we like to do is we like in the models, when we want to understand how everything's working, we want to put them into groups, different groups or functional types. And in the original ones, it would only be phytoplankton, zooplankton. And now we have got mixoplankton, and functional type wise, you can say broadly 2 types. But then again, within the non-constitutive, we've got 3 more functional types. So So because of this, then in 2019, so this work's been going on for decades, and in 2019, because we found that they were so different, there were so many functional types, we proposed that the term mixoplankton is used for these organisms instead of just calling them photo-phago mixotrophs or anything. So we have got mixoplankton, which are protists capable of obtaining nourishment via their photoautotrophy. So that's phototrophy, osmotrophy, and phagotrophy. And it was published in that journal. So now with the new functional types, when we look at our food web and see what happens to it, this is what we see. We see that at the base now, we have got mixoplankton, which are producing and consuming, and say there's an arrow between the mixoplankton and the multicellular metazoan grazers, because we know they can ingest what would have been their predators, and then we've got the phytoplankton and the zooplankton. And when we have this, and what we wanted to see is how many of what do we know about how many species are there, who are they, what sizes are there, what groups are there. And we started looking at it, and we ended up developing a new database, a mixoplankton database, where we've got about 450 species in there, and we're finding more out, for that's the last count. So this, and when we looked at the different phytoplankton and zooplankton, what we found, there's a lot of what is known as iconic phytoplankton are actually mixoplankton, and various exemplar zooplankton are actually mixoplankton. So we have got a revised concept. We have got a database. The question then is, what does it mean in terms of biodiversity in nature and biogeography? So biodiversity, comes back to the evolutionary pathway when we looked at it going both ways, and we look at the eukaryotic tree of life, and these are the different types. So this is only the eukaryotes, and you can see how there are different types of mixoplankton in the whole tree. And this is because, and you can see there's lots of different CMs within the same group, CMs, NCM, and that's because they would have had this evolution going both ways. And at some point it would have evolved to different types. To put this in context, this is where we have got the classical phytoplankton, which are the diatoms. This is where we got the plants, the land plants, and the fungi. And that's where we got all the animals, including us. So when you look across the eukaryotic tree of life, there's a lot more protist plankton and mixoplankton, or protist and then mixoplankton in them, compared to what our diversity, or the animal diversity. Size-wise as well, they are single-celled organisms, but the size is amazing. The range is amazing. They go from really small, which is pico, through to, so here you've got the 1 millimeter, through to macro. So the forams we're talking of, they are in millimeter sizes, the forams, the radiolarians. And that is only the size of a single cell. A lot of them can form colonies, and then can form chains. So for example, I don't know, here in a few months, in the summer months, we get the foam in the sea. I don't know if you get the foam on the sea. Yeah, I can see some heads nodding. Yes, that's a mixoplankton. That's Phaeocystis. So this time of the year, they will be single cell. They're at least in our waters, they're swimming around single cells, but by summer, they will then start forming colonies, and that's what we see as foams. So that's another size. So that's not within this. And biogeography. So the next question is, of course, we have got different sizes, different shapes, and everybody else. And if you're trying to tell the scientific community they're important, where are they? Where can we find them? So we did a study of biogeography across the ocean, global ocean. So this is the non-constitutive maps. So the non-constitutive are the ones which need to acquire phototrophy, either from specialists or generalists. So these are the generalists. So you can see, so in the map, the white zones are the ones where nobody has looked for them, rather than not found them. And the blue zones are where people have looked, and you can see the dots are where they have found them. So these need to top up regularly to keep it going. Then these are the plastidic specialists, which need to top up, which can go for weeks, don't need as regular top up. So these are our Mesodinium and Dinophysis. that the three group of three, and then we have the ones which have the PSS and ESNCM, which are then the symbiotic ones, which are the ones which are harboring symbionts. So you can see they're absolutely everywhere. Then we looked at the constitutive mixoplankton biodiversity and biogeography. So here on this axis, the x-axis, we've got the different types. So these are functional types. So they are prasinophytes, dinophytes, rhaphidophyites. And then on the y-axis, we've got the size within the nano size class. So these are all between 5 micron to 20 micron diameter. So there are a lot of the harmful algal bloom species fall within this size range. A lot of the good food, which link up the primary production through to the fisheries, fall here as well. And again, we find the missing maps are because we don't have any organisms within this group in that size range. So that's why the white blank spaces. And again, the white spaces in the maps are because people haven't looked for these organisms, and the colors show you where they are. So the dinophytes have been looked at quite a lot because a lot of the harmful algal species form in this group. And now looking across all the 4 functional types on the x-axis. So we've got the generalist, the clasteric, the ESNCM, and the constitutive. So and then on the y-axis, we've got the different sizes ranging from pico nano, so which is from 0.2 to 10 micron here, and then going through to macro, which is over 300 micron. We can see that again, they are absolutely everywhere. But they are everything is not everywhere all the time. So this map shows us where they are available, but it doesn't mean that they are there. If you go out now, you'll find all of them at the same time. There is seasonality in when they turn up, where they turn up, and what different climate factors which drive their bloom conditions. So what does this mean for the plankton food web and the mixoplankton paradigm? With respect to the mixoplankton paradigm. So most of the climate change, well, actually all of the climate change and the fisheries models have used this framework from the 1990s. So this is called the NPZB model. So the N is for nutrients. So we've got the dissolved organic matter. Then we've got the dissolved inorganic nutrients. So this is ammonium, nitrate, phosphates. And then the P is your phytoplankton here. Z is a zooplankton. So Z is a zooplankton, protozooplankton, and the zooplankton. And then we have got B, which is the bacteria. The higher trophic levels, so the zooplankton here are the jellies, the krills, the shrimps, and all those, and juvenile fish. And the high trophic levels are adult fish and whales, sharks, etc. will fall in here. There's the physical processes, which is light, mixing temperature, turbulence, etc. So this is at the base of all models across, looking at climate change, looking at ecosystem dynamics and fishery management. So this is under the old paradigm. When we put the mixoplankton in this, we find there's a lot more of interactions here. So they are taking up dissolved organics, they're taking up dissolved inorganics, they're eating bacteria, they're eating phytoplankton, they're eating protozooplankton, sometimes they're even eating zooplankton. And this is just with mixoplankton as one group. So if you then consider the different functional types, think about putting them in. This is going to get more complex. And this is also not looking at sizes. So there's within each of these, there'll be size categories, and the interactions will be different. So if you can imagine the complexity, the fun doesn't just stop. And why does it matter having all this? Because the question then is, okay, it's fine. We found a new, all these organisms which have got all these strategies are everywhere doing all these different things. So what does it actually matter in the grand scale of things? It matters because in terms of biodiversity, because depending on what the conditions are, what the nutrients are, what the climate is, the temperature, turbulence, etc., we can have good mixoplankton, which are going to go and support all the fisheries and make money for the country, the people who are working in the sector, and also look after all the high trophic levels, keep a healthy ocean. It's healthy ocean, healthy system, great outdoors and everything. But under different conditions, under bad conditions, you can get the harmful blooms. So the dinophysis we looked at, which steals from the Mesodinium, with the straw-like, those are the ones which end up with shell fisheries closures. I think you have them closures on the west coast. You have oyster farms which are closed now and then, hatcheries. So those would be dinophysis, could be heterosigmas. I know Chesapeake Bay also have a lot of harmful blooms as well. And then, of course, it's fish kills, and then dead fish, bad smell. It affects lots of things. It affects, I know in Australia, they've had such bad harmful blooms, and the smell, it's affected house prices. That's what I've heard from a colleague. So that's why it's very important to consider the diversity, to consider mixoplankton, and not just stay with the phytoplankton, zooplankton dichotomy. So the big question is how and why were the mixoplankton missed out? How come nobody has, it's taken so long, 2019, for this term to even appear in science? It's because the traditional plant animal, the phytoplankton-zooplankton dichotomy, has been long established. It's been the bedrock of ocean science, of marine science, marine biology, ecology. It has driven everything, all textbooks and everything. Mixoplankton, the organisms have been known for decades, but they have been considered as weird and wonderful, sort of outliers rather than mainstream. And this has resulted in field work, culture maintenance, laboratory experiments, and simulation modeling, all optimized for non-mixoplankton. So they're all optimized for phytoplankton. So for example, the field work does not take into account the fact that mixoplankton are quite fragile. So the different types of capturing or collecting plankton does not then collect mixoplankton, or they may turn them into a mush and you don't know what's there. They also, when they're preserved, the different preservatives used are not suitable for mixoplankton. They're all, again, suitable more for phytoplankton or zooplankton. Culture maintenance, a lot of the lab experiments or experiments done with the data for which are used for modeling, etc., are based on the plankton which have been isolated and are maintained in culture conditions, which generally means no food. And what we have found, if we keep the mixoplankton without food, over time, it loses the ability to eat. So it evolves to have a lower growth rate, and it forgets to eat. So as a result, if you're working with something from a culture collection, so there's Bigelow's, we've got the culture collection in USA, we've got there's Roscoff in France, we've got CCAP in Scotland. They're all kept as phytoplankton, so they have not been given food. So over time, they have lost the ability to eat, and this is why we have missed out. So what needs to be done? What have we been doing? All research methods need to be revised. So research methods need to take account of the fact there are mixoplankton which are eating and photosynthesizing. And this is a problem because a lot of the predator-prey experiments are based on the fact the phytoplankton don't eat and the zooplankton eat. So it's either done in full light or full dark conditions, depending on what is being studied. And now you think of mixoplankton using both light and dark, eating and photosynthesizing. So there needs to be different research methods. We also need to change the scientific outlook. People need to start thinking, we all need to start thinking of mixoplankton as part of the community of plankton, of algae, not just something which is on the side. And once we start doing this sort of thing, it will be fun because we can have lots of new discoveries, because suddenly we've opened up a whole new world. And one of these things is what we found is this cosmopolitan Procentrum genus, which you can see occurs everywhere in the world, and there are lots of different species within there. So I've picked two of the ones which we have been studying, Caudatum and Pervagatum. And what we have found, these these have traditionally been labeled as phytoplankton. So they have been labeled as primary producers everywhere, ubiquitous primary producers. But we've found that they make mucus traps and use these to capture prey. So this is a schematic of how we think it does. So you've got the procentrum here. So for plankton, when they're doing photosynthesis, they produce mucus. So that's why, they're always producing mucus and they're releasing mucus. So that's what we see here. And then at some point, with some random swimming around, it's not like we haven't seen an exact obvious way it swims around. It will swim around and it forms this mucus trap. So you've got the procentrum here, it's got its mucus trap and it's got its prey. So it's got these big holes you can see. So what it does, activity, it draw with its flagellates, it draws the water in. And as it's drawing the water in, it's drawing the prey into its mucus ball, and it's trapping them here. So they get stuck on the insides. And then you have got the smaller holes where the water can escape. So in essence, what it's doing is it's having this constant flow of in and out to bring in the water and prey with it. The prey gets stuck, the water goes out. And these traps are made, from what we can see, they're made and replaced every day. So it's not a matter of it stays in one. And what happens in the discarded traps, along with whatever it's captured, including unconsumed prey. So everything it captures, it doesn't eat. So there are parts of it which then sinks to the bottom and then it makes its own again. So we've got a video here of, so you got a procentrum, you've got the trap. So it's got all the little bits you can see in there. Those are all prey. Here we've got three traps. So one procentrum. So in each trap, there's only one procentrum ever. And Urban, who made these videos, here you go. Here, you can see them again here. So it's loaded with teleolax, which is the poor teleolax, everything eats it. So this is the one which the Mesodinium and Dinophysis need. So you can see them. There's a swimming. They've got the traps here, and what we did find, Urban did find that once a procentrum went in by mistake into another trap, and it jumped out quickly. So the traps are somehow they know their own trap. They have got, obviously, they have got their own escape mechanism. And here we can see it. So if I pause this. So you can see, so it's got two flagella. So one of the flagella here, it's parked itself in the trap. And this is another flagella up here. So it's moving it like a helicopter blade. And that's how, and you can see it's dragging things in with that. So it's rotating, it will drag these things in. And then what will happen is, and then it will stop and it will go around eating. Eating, and then when it's done, when it gets too heavy or something, it leaves it and goes and makes another one. So there are lots of videos for this. If you're really, really interesting, they're all free. You can go and have a look at, I can spend hours. So Urban obviously has spent hours doing these. So so there are there's lots to find, lots to do, lots of new things to discover. It's only the start. This is literally the start of the story, Mixoplankton story. So to summarize, what we have found, our research has found is that mixoplankton are not outliers. They are key contributors to biodiversity, their biogeochemical cycling, and ecology. So there's wide diversity. It's not just one mixoplankton. There are different types of mixoplankton, different shapes, different sizes, different parts of the world, different climate, different things making them grow and bloom, etc. And in essence, we've got Pandora's boxes open, and we've got lots of these things coming out, lots to discover, lots to do. So anybody interested, this is the time to come into marine science and find more about the mixoplankton. So thank you.
Thank you so much. I'm sure you all can see why I found her work so incredibly enthralling. We've been teaching food webs and food chains. Oops, so sorry. The joys of being in a school. You got to hear the bus announcements. We've been teaching it the same way for so long. We've got phytoplankton, zooplankton at the base of the food chain, and here there's this whole other thing that we've known about for 7 years and yet has yet to make it to any of our textbooks and into our classrooms. Does anybody have any questions, or did any of your students have any questions in your rooms?
Do you want me to stop sharing?
You can,
Or
so we can
unless
see
people
each
want
other
to
maybe a little better.
yeah, if people want to have
Yeah.
questions from
You can
different.
also put them in the chat and we can go from there. It's up to you all. Yeah, Lisa.
Well, this was clearly very interesting. It
Thank you.
sounds like dog eat dog in this world of mixoplankton. And I really had no idea about this at all. But when you showed the brief video clip of, I think it could have been a dinoflagellate. It's here in your Pandora's box in the upper left-hand corner. And when it was being eaten, yeah. So
Yeah.
when it was being eaten by three other, I don't know, plankton,
The mixoplankton as well, yeah.
right. So they seemed very aggressive. And I was just wondering if that's because in that scenario there was so few prey that once they sense the prey and latch onto it, they have to have mechanisms in order to be able to eat quickly and not let the prey go. So I was just wondering, about all of these things in the toolbox, that the plankton has, and or it's sort of similar to when you go down to the depths of the ocean. The fish down there have all these very interesting um, techniques and pieces of equipment and abilities to merge with another organism, so that they don't get away from each other. And so I was wondering if there was something similar going on here within the mixoplankton.
Yeah, they they are they occur in patches in the ocean, and there's also what is called allelochemical. So they're releasing different products, and that attracts prey. So, and there's also different types of feeding. So that one where we had this tripos, which is a mixoplankton itself, being eaten by the three smaller mixoplankton. Those do engulfment, so that's why you saw they like eat like that. So goes. But then you have got the others. There also the other one is a peduncle feeder, and there are lots of different ways they will feed. So there are different types of feeding mechanisms, and then there are of course different times they occur, different places they occur, but they are chemical cues. It's been known chemical cues has been known about just attracting, attracts bacteria, attracts lots of different things. So yeah, absolutely. They don't see it. There's more chemicals rather than vision because obviously no eyes there. So.
Thank you.
One of my students was curious how you first got interested in plankton. Why plankton?
Why plankton? Oh,
Mhm.
it was accidental, really, because my background, I read botany and chemistry in university, and then undergrad, and then I did a master's in ecology epidemiology, and that's when I got interested in modeling, and I was modeling in epidemiology, and that's what I thought I was going to do. But while I was waiting for funding, PhD, as one does, there was this job up in Scotland in the Scottish Association for Marine Sciences. It was what was attractive for me was the first modeling position which did not need a PhD, and it was modeling these things called microzooplankton. Right? I thought I'm going to apply because it doesn't need a PhD, and microzooplankton, I'll read it up. It's, I know a bit of phytoplankton from botany. So I applied, and surprisingly, I actually got the job. And that's how I got hooked. So it was totally accidental. I never went back to epidemiology. After that, they asked if I want to stay on and do a PhD. So I got funding to stay, to carry on my PhD work in SAMS in Scotland. And yeah, so here, I'm still here. So it was never planned. It's just, and mixoplankton. So I started with zooplankton. Because when I started doing my PhD, which was on zooplankton, the protozooplankton, there were no models which were connecting the phytoplankton to the fishery. So that's how I started looking at the quality and quantity of prey and how that affects the zooplankton quality for the fishery sector. And I came across these terms like these organisms which are stealing, photosynthesizing, mixotrophic, etc. And I thought, oh, that's interesting. So I got in touch with Diane Stoker, who's in Hornpoint, who's an emeritus professor now. And she's done a lot of work on this. And I asked her about this. And that's when we started discussing, talking, and then I started applying. We applied for lots of grants. I applied for lots of grants. We said, no, no, they're not important. They're not important. You don't need to know about them. Everybody knows whatever little bit we need to know. And then I got lucky. We got a grant from Leverhulme Trust to put together a networking, international networking grant. And that was bringing together people from all across the globe. So basically from America through to Australia, South Korea, anybody and everybody who'd worked on mixotrophic plankton, mixoplankton. And that's where we started putting these things together, finding out how much who knew what, making the data. So that led to the database and all this work. So yeah, and then I've been lucky with different funding. So we just carried on collaborating. And so that's how I got in, and I'm here.
That's amazing. And I think that's such an important story for students to hear, right? Because they all think they have to know by 13 exactly what they're going to do. And they seem to have this belief that they have to have their life planned out. And none of us end up where we thought we would be when we were a teenager.
Yeah.
Anyway, so I think that trajectory is really important. I'm like an hour or two from Horn Point. So the fact that I learned this from a researcher at Cardiff University while in Scotland, when the state right next to mine has another expert in it, is another beautiful thing about the global part of our communication and science. So
yeah, I also like the way you connected the plankton to climate and seasonality and temperature changes. I think that's really very important because that's something more concrete that people are used to hearing about and understand a little bit better. And so if, for example, if they see a red tide or if they hear about an alert not to eat mussels, this all has to do with plankton on some level. And so it's probably a great entry point from what they're seeing out in the waters into what you're actually studying and talking about.
Good point.
Yeah, there is, Lisa Campbell, and she's retired now, but she's in Texas, and she set up this program called Toast, like bread toast, and that has been monitoring the dinophysis because they had serious problems with dinophysis blooms and affecting shell fisheries. So they've been doing that. So they've got this fancy equipment, but it's in some ways it's easy when it's only one type you're looking. So they're looking at the shapes. They've done it's not easy, but they've done the machine learning and etc. for them for this program to pick these up. So constant survey and then say, okay, if it's this, this is we've got too many in the water. All these things have so much per liter or per cubic meter. And when it reaches that, so there's red alerts going. So yeah, it's climate is also a lot of eutrophication, nutrients. And it's a balance of nitrogen and phosphorus, which changes a lot of my understanding from what I've heard is that a lot of the Chesapeake Bay blooms are because of that change in balance of nitrogen and phosphorus in the water.
Yes. Yes, we have a lot of eutrophication for sure in the Chesapeake Bay. Well, I think if there are no other questions, we just thank Dr. Mitra for her incredible expertise and time and for sharing it with us. I'm looking forward to figuring out how I'm going to incorporate this into my classroom for sure, and brainstorming with some of the NMEA members in the webinar next week.
Yes, thank you so much, Dr. Mitra. And
thank you.
Thanks everybody who was able to come today.
Thank you, everybody.
Thanks for coming on so late for you.
No, it's okay.
Bye, everyone.
Next week's later.
I know.
Yeah.
Thank you all. So grateful.
Thanks.
Thank you.
Thanks. Bye.
Bye.